Hi, this video is a debrief on the enzyme quiz that was given towards the end of term 1. Okay, so let me start. The first question shows you burning of a particular food sample. So there are 4 samples that is being burnt. Okay, and of course the question asks which one has the greatest increase in water temperature. Alright, so this is the energy question. The energy is of course locked in the nutrients in the food. Energy is found in carbohydrates, energy is found in fats, and energy is found in proteins. Okay, fats has the highest energy in theory because fats is a rich source of energy, and fats is a long-term storage of energy. Okay, so fats contains the highest energy. When I burn a food and the food releases a lot of heat, it means that the food has a lot of fats. Okay, does it have a lot of starch and proteins? Of course, but fats is the one that will show the greatest increase in temperature because it's heat energy. So if you take a look at S, right? S, which I have highlighted already over here, you can see that S has the highest bar for fats in these 4 foods. And so the question asks, right, greatest increase, and so the answer is S. Okay, very straightforward, very simple. All right. The second question asks about the elements which is found in a molecule of starch. So remember, elements is not elements is like elements. Okay, so it's not a compound, it is not a molecule. And so for starch, right, the elements contained in starch are carbon, hydrogen, oxygen. The elements in fats is also carbon, hydrogen, oxygen. The elements in protein is carbon, hydrogen, oxygen, and nitrogen. Okay, so don't get your elements mixed up with molecules and compounds and whatever. All right. So for part C, fats has been linked with many health problems such as obesity and heart attack. So despite this, blah blah blah, a balanced diet is needed to include fats. Okay, so basically they ask why fats are needed in a balanced diet. So it is an indirect question on what are the functions of fats, why fats are so important. Okay, for a balanced diet. In this context, right, because of the word balanced diet and all this stuff, right, your answer should be more tuned to animal cells functions. Okay, so for fats, it is mainly animal cells or plant cells. But if this is a carbohydrate question, right, then don't write plant stuff at all. Okay, write stuff that is closer to the context. So number 1, right, fats is a rich source. This is not in your notes, but I would prefer that you write the word rich in the answer. Okay. And then of course it's a long-term storage of energy. So this is a reason why. Number 2, fats is a good material against heat loss, which I mentioned in class before. It is not just against heat, it must be heat loss. Okay, it is to prevent our body from losing too much heat. So the word heat and the word heat loss is not the same. So just remember to write properly. Okay, solvent for fat soluble vitamins and some hormones. It is a solvent. It is a good solvent for some of them. And number 4, the fourth one, it is essential in the formation of cell membranes, or you can say it's a component in the making or the formation, the synthesis of cell membrane. Okay, now I think these 4, which I already mentioned here, these 4 functions are good enough. All right, the 5th function, right, that we throw in extra, right, is for water conservation, and this one is in the notes, of course, but this is elaborated, and the one in the notes is more like a short form thingy. Okay, so please write properly. All right, when you study functions, I wrote over here, please write in full. Please don't anyhow write. And yeah, okay, if you can elaborate, please elaborate. So question 2, right, is an enzyme question. So I have an experiment over here. It is the original amount is 6.5 grams, and pepsin is used in this. And again, you should know that pepsin breaks down proteins to polypeptides. Okay, and proteins is found in egg whites. So pepsin is the enzyme, protein is the substrate, polypeptide is the product. Okay, at pH 2, so it is pretty good because pepsin works in acidic pH. And this time, right, the range is from 10 degrees to 60 degrees Celsius. Okay, as you can see that it increases from 10 to about 40. The remaining mass, right, is becoming lesser and lesser. It means that there is more digestion. Okay, so this graph is inverted. It's not the typical the one that you see in the notes because of the y-axis. So you must be careful. All right. So the first question: describe and explain the effect of rising from 10 to 30. So there are two common words over here. Okay. Number one is describe. Number two is explain. Oh, so I mentioned this in class, so I'm going to repeat this one more time. Okay. If you are given data such as a graph, a table, a bar chart with numbers, that's why we call data, and the command word is describe, right? You just have to describe the trend. Okay, so there is no need to go into the explanation because it will come later. So when you see describe the effect, right, in this case, it actually means just to interpret the numbers. So you can see over here in the first line, from 10 to 30 degrees Celsius, the undigested egg white, which is the y-axis, right, decrease from 3.5 gram to 1.0 gram. You can see, right, that the data on the x-axis is listed. The data on the y-axis is also written. Okay, and of course the trend is a downward trend, which is a decrease. So this is known as describe. Please be careful, okay, when you answer questions, and the command word actually tells you how to answer. And normally describe data question, right, is range from it's about 1 to 2 marks. Okay, but yes, some students, they don't interpret properly the command word, right, and they straight away go into the theory, which means that they are not even answering the question. Okay, so that is explain, right? Theory is explain. Describe is describe. Okay, so I just want to let you all know that for this unique question, right, because there's a graph and there are data given and the command word is describe, so you just have to input the data. Okay, naturally the next question is to explain why. So the why part, right, you can see over here that I've underlined so many keywords. It's actually all found in the theory for enzyme reaction with temperature. Okay, so at 10 degrees, right, let me go through. At 10 degrees, the enzymes are considered inactive. This is a keyword. Please write properly. As I increase from 10 to 30, as I slowly increase the temperature, all the molecules are, so please write enzyme and substrate molecule. It's not just enzyme. Okay, all the molecules in the solution increase in kinetic energy, means they move faster and faster. And when they move faster and faster, right, they will collide with each other more. And even though I keep colliding with each other, I might not be able to fit into the active site of the enzyme. And so the collision, right, must be effective. Okay, so there must be more effective collision between the enzyme and substrate molecules. And if this collision is effective, I can form more enzyme substrate complex. Alright, if I can form more enzyme substrate complex, means I can form more product molecules, which means the reaction works and the reaction becomes better and better and better, or the reaction increases. Okay, so this is how you explain enzyme theory with respect to temperature. Okay, you can see that the keywords are all down and you cannot anyhow write it in a, in a layman term point of view. Okay, so please go and study this. It is a very straightforward question, but it involves a little bit of effort. So in the second experiment, the pepsin is incubated at 100 degrees Celsius, very hot, and everybody knows that. And even though it's cool, right, it will be denatured. Okay, so the first one, draw a line, which I didn't show in the diagram. It is a straight line at 6.5, which I already mentioned to you already. It's just a straight line. Okay, but this one, explain what happens. So the keyword is denature and no other words. Paraphrase from 100 degrees Celsius, please. And so write denatured by the high temperature. And because these 2 marks, you have to explain. So from here on, you have to move on. Okay, so during denaturation, right, the active site is altered, the active site change, unable to bind with the substrate molecule, unable to form enzyme substrate complex, which means it's unable to form products. correct? Yeah. So basically just have to explain deeper for denaturation. Okay, because of the 2 marks. If it's 1 mark, I think denature by high temperature is good enough. If it's 2 marks or 3 marks, then you have to elaborate more. Okay, and they are all explanation, and again it is enzyme theory. Okay, the last question or the last part of this particular quiz is basically a repeat of the first question in worksheet 1. So you have a series of shapes, okay, and each of the shapes, right, represents a nutrient, a different nutrient. The question asks for protein makes up of 6 basic units. Basic units, right, means amino acids. So because it's a protein and it's made up of 6 basic units of amino acids, you just need to draw these 3 diagrams. These 4 all cannot draw. Okay? Even if you repeat, it is perfectly fine. And so your 6 amino acid diagram must show that they are all joined. And this is also legit because they are all amino acids. Okay, so you can choose any combination, but you must choose 6. And so you will get your mark, which is the final 1 mark. Okay, so yeah, that's it. Okay, so this is my debrief for the quiz. Please ask me if you have any questions when you see me again or when you do your revision. Okay, thank you. Bye bye.